Medtronic Technologies impacted more than 72 million people in the last year, equating to 2 people every second. Harnessing the power of technology to take healthcare further, each technology has unique benefits designed to serve patients. The goal of this program is to get closer to the patient and delve into the challenges and impact of each technology in practice. This is the Medtronic MedEd Learning Experience. The INVOS monitoring system should not be used as the sole basis for diagnosis or therapy and is intended only as an adjunct in patient assessment. The contents and conclusions of the following programs are solely those of the speakers unless otherwise cited. The speakers are responsible for all content and any necessary permissions. The speakers receive funding from Covidien LP, a Medtronic company, for this speaking engagement. Medtronic's medical education programs are offered to provide attendees education on FDA-cleared indications and use of our products when applicable. For this segment of the series, a discussion on the utility of the INVOS system. How is the INVOS algorithm developed for the pediatric population? To help answer this question is Eli Coleman, Senior Research Manager at Medtronic.
In this next section, we will discuss the INVOS algorithm that is used and developed specific to our most vulnerable population, the pediatric population. During development, what's currently the adult INVOS algorithm was used in a study with pediatric patients. Finding was that the measurement showed a significant bias, greater than 12 RSO2 points on average, with this segment of the population. You can see that with the graph on the left. The regression line is shifted down from the 1-to-1 line that would show perfect correlation. However, the trend of regional oxygen saturation, or the accuracy of changes in oxygenation, was still excellent, as seen on the right-hand side graph. There are many potential anatomical and physiological differences in pediatrics that may contribute to this offset error, including head circumference, skull thickness, depth of tissue, concentration of other non-hemoglobin chromophores such as water or protein. And it may be that just a few from this list are significant factors, or maybe that all of these factors stack up together to create the error seen. Regardless, given the number of likely factors, it's not surprising that a difference would be seen. When looking at the error of RSO2 across pooled populations of adults and children, an interesting trend is highlighted. The graph on the left has weight on the x-axis, which correlates to age, and error is plotted as bias on the y-axis. As the fitted line shows, error begins to become increasingly large the lower the weight or younger the subject. It was determined that 40 kilograms was a reasonable cutoff to implement a different algorithm. After development of a less than 40 kilogram algorithm, the same data was run through the same analysis and shown on the right-hand side. Now the trend line is noticeably flatter and overall closer to the zero bias point across the weight range, which gives us a strong indication of an effective algorithm change. Another notable source of error due to differences in physiology is skin pigmentation, which will be covered next.
Please tune in next week for a new segment from this series wherever you find your podcast. This is the Medtronic MedEd Learning Experience. Thank you for listening.